When the war was just on the verge of being declared, I came back from Belgium to Turin, and there I found myself in the impossibility of doing any work at the university because of my racial position. I don't know how you would call it, I don't care about. You know, race do not exist. As it had been said, race do not exist, but racists do exist. So I was at that time already a medical doctor. I took my degree in 1936. So I had to abandon my position. I could not go anymore to the university where I've been working on neurobiology from 1936. Even before 1935, started working with Professor Vicentini. In this condition, I could only do two things: either move to the United States, and this I would have liked, but family did not want, and I did not want to separate myself from my mother and brother and sister, whom I loved very, very much. For this reason, I decided to continue my work in my bedroom in Turin. At the time as the war started in 1940, just after the law, which had been promulgated, a racial law in 1938 and activated in 1939, had been put out of work. I could not do any more medical work. I was a medical doctor at the time. I could not do any scientific work. I established a laboratory room in my bedroom, in my laboratory, small laboratory in my bedroom, continuing to work, which ever since 1935 had been working on at the University of Turin with Professor Vicentini. When the war was declared, June 1940, it became difficult to continue to work in Turin, and we moved to the country near Asti, small village close to the city of Asti. And there I continued my work. Even it was a tremendous vicious campaign against Jewish people, but I didn't care about, you know. At the time it was more voice and really very vicious, but no personal attack. So I continued to work, completely ignoring what was happening around me. I didn't care. I read the article by Victor Hamburger, the 1934 article, while I was going from Turin to this small place on the country. But, as I mentioned, I'd been interested in neurobiology, experimental neurobiology, long before then. That is, from 1935, I'd been working at the university. At the time I was a medical student, the last year of medicine, when I first started working on neurobiology, and particularly on the chick embryo, without knowing anything about Victor Hamburger. This was with my friend, who later on passed, a very good physiologist. We worked on the neurophysiology of the chick embryo, how the movement started. Came out a long article during the war, article in Switzerland. It was the article which came to the attention later on also to Victor Hamburger with the other. I was not in agreement with Victor Hamburger about the reason why, when you cut the limb bird in a chick embryo, the sensory and motor cells just undergo atrophy and degeneration. He thought it was a phenomenon of induction, lacking induction. I personally didn't believe so, and in this article, which he read and was very interested in, I decided, and Giuseppe Levi is also co-author in this article, that the cause of death was not a phenomenon of lack of induction from the periphery, but simply nutrient lacking. That is, by taking away the limb bird, the nerve cells, sensory and motor cells, which normally innervate the limb bird, were lacking something, a nutrient. I didn't know what kind of nutrient was it, coming from the periphery, so it was an entirely different interpretation from the one Victor Hamburger had been given. Victor Hamburger was a student of the great embryologist Spemann, and just thought that the phenomenon was a lack of induction. I didn't believe so. In 1946, I had already my position as assistant of Professor Giuseppe Levi at the university. Giuseppe Levi received a letter from Victor Hamburger, a very beautiful letter. I still have this letter. And Giuseppe Levi called me and said, here is a letter of invitation to you. Will you accept it? The letter was written in German. Victor, naturally, was German, and Professor Giuseppe Levi was very fluent in German, so he translated to me the letter. Professor Victor Hamburger wrote to Giuseppe Levi, I know that a young person, a young lady working with you, has been working on the same problem which I have been working on ever since 1934. Would she will accept to come for 1, 2 weeks or even months to work with me in Washington University, where he was chairman of the Department of Zoology. Professor Levi asked me, do you want to go? But at the time I had some doubt if I wanted to go, and they delayed until 1947. I was very excited by the letter, but I did not really want to immediately move away. When I received the invitation by Professor Victor Hamburger and took with me, it is perfectly true what you said. The special thing is my experience and really particular gift, I must say, to work with microsurgical instruments which I had prepared myself. They were different from Spearman's instrument. As perhaps you know, Spearman and Victor Hamburger worked with glass needle, which they fabricated. I preferred just normal needle, the needle which you use for sewing, and I transformed this needle on a mold in a microsurgical instrument. And I was very, very capable in just implanting, transplanting piece of the embryo. And this had been doing before even going to the United States. Besides this particular competence in working with microsurgical instruments à la Renaissance, you can see, I became very competent in tissue culture. Professor Levi was a great scientist and a great man. His merit was not only to be an excellent person and very good friend of mine ever since up to the end of his life in 92. But he had also introduced me, he was a pioneer in the tissue culture technique, which no one knew in Italy. Carrel had written his famous book, and of course it was well known, the technique in the United States. But I was not very familiar. When I went to Washington, I saw that no one knew about this technique. I was instead very competent. We spent 2 years with Giuseppe Levi before being thrown out from the university and working in tissue culture. So this was the special gift or special instrument, if you want, which I took with me when I arrived at Washington University. That is the competence in surgical instrument and in the Cahal technique, which no one knew at that time, and this was what I took, and what was the reason later on for my discovery. That is, I was very competent in the study of the neuroembryology, because for 5 years I've been working on neuroembryology with the Cahal de Castro technique. I was very competent in surgical instrumentation, which I prepared myself à la Renaissance type, and I was also very competent in tissue culture, 3 things. Immediately after I arrived, I took back, and we worked with Victor Hamburger, the problem, how it is that when you take away ablation of a limb bird or transplantation of an additional limb, you have, in the first case, a decrease of number of sensory motor cells. In the opposite case of transplantation, increase in sensory motor cells innervating the additional limb. And came out to work together with Victor Hamburger, where he recognized that I was correct and he was not, that it was a matter when you ablate, when you take away, cut a leg, what is missing is something from the periphery, which is necessary for the limb bird, for the nerve cell to grow. It was not a matter of induction, it was a matter simply of lack of nutrient. And this was the paper which came out with Victor Hamburger at the time, one of the papers, because another paper which I wrote all by myself, it was on apoptosis and cell migration, which came out in 1950, and it was quite a discovery. But after having just done this discovery of, I didn't know it was apoptosis, programmed cell death, I knew that some part of some sensory ganglia in the cheek embryo undergo massive cell death, and also massive cell death occur in the cervical level of the embryo. And this I described as an occurrence. I did not know at the time it was a programmed, it was a physiological process, not consequent to ablation of a leg. But the ablation of the leg also brought about cell death. And even now the situation is not very clear, because the programmed cell death is just what happened, and we know after apoptosis description and the gene discovery about apoptosis, there's been a tremendous amount of work recently. However, they recognized to me a sort of priority, because I was the first to describe this massive cell death under normal conditions. Later on, when we make the surgical ablation of the leg bird, also we have cell death. And at that time we were not very clear that it was the same phenomenon of apoptosis. So it was very, for a long time, practically ignores the problem, what is going on normally, physiologically, which is massive programmed cell death, and what is not physiological, but experimentally produced or surgically produced or accidentally produced lack of periphery. That is something which is lacking, and just is a question which happens at the level of gene of the cell programmed cell death. One morning, winter morning, I believe it was 1950, Victor Hamburger called me to this laboratory. The two laboratories were one close to the other, and said, I received a letter from a former student of mine by the name of Elmer Bucher, and he wrote about a curious experiment he performed. He transplanted a fragment of a malignant tumor of mouse in the side of a cheek embryo 3 days old. And what he found, and described in this article which came out in 1948, that sensory fibers from the embryo entered into the tumor, and 3 days later, when he checked the normal histological technique, he found that the ganglia were slightly larger than the opposite ganglia which were innervating the limb. The person, Elmer Bucher, thought that in the tumor was some histochemical property which attracted the sensory fibers to enter into the tumor. When I read this article, I found the effect extraordinary, and I was terribly enthusiastic about repeating the experiment, and said so to Victor Hamburger. Victor Hamburger agreed with me, but then he left for Boston, where he was doing some experiment, a genetic experiment. And I received, at the time that he was gone, the mouse, many mice carrying this tumor, mouse sarcoma 180, which is a mammary carcinoma, was at first. Then it became called sarcoma because its growing is not typical of carcinoma but of sarcoma. While Victor Hamburger was in Boston, I made the first series of experiments, and to my great astonishment and pleasure, I found out that not only what Elmer Bucher had decided and described was correct, but it was far more than that. Not only sensory fibers entered into the tumor and branched in. He had not seen it because he did not know the tissue culture, the Cahal technique. He was not familiar with this technique. I saw that the fiber branched very regular without established contact with the cells, but just branching between the cells. And also I found out that not only sensory, but also sympathetic fibers entered into the tumor in even larger number than the sensory fibers. And what was to me extraordinary was that not only was the ganglia were enlarged very mildly, as Bucher had described, but were 6 to 8 times larger. He had only found a minor difference between the ganglia innervating the tumor and the ganglia innervating the leg. So I told to myself, this effect is a tremendous effect which has nothing to do with the regular effects of periphery. The tumor must be something extraordinarily important which makes the sensory and sympathetic fibers grow much larger than normal and branching into the tumor, not the motor fibers. When Victor Hamburg came back from Boston, he joined me in the article Levi-Montalcini and Hamburg in 1951, which describes this effect. At the time, I still believed that this effect was restricted only to the sensory and sympathetic fibers which had reached the tumor. Victor Hamburger did not follow very close. He was a very intelligent and very highly educated person, but not particularly familiar with neurobiology. I continued this work and described an effect, extraordinary effect, later on as the New York Academy of Science, December 1952. And there I described something completely new. I say not only some large number of fibers enter into the tumor from the ganglia adjacent to the tumor, but all the sympathetic ganglia of the embryo, which are even very distant from the tumor itself, are enlarged. So I said in this article, which became a very classical article, 1952 New York Academy of Science, I said, I do believe that this is due to some substance which is released from the tumor and reach all the cells which are receptive to it. And this was my first discovery. So I say, what about if I transplant, in the same article I wrote, the tumor not into the embryo, but far away from the embryo in the coriolantoid membrane. As you know, the coriolantoid membrane, which envelope the embryo and the entire eggs, is connected to the embryo through the circulation. That is, the blood cell fibers, the circulation, blood cells go from the embryo to the yolk and take food in. So I transplanted a small fragment of this malignant sarcoma 180 or sarcoma 37 away from the embryo, and there is the same effect. So I was enthusiastic about it, because I said to myself and also to Victor Hamburger, I said, I have discovered something entirely new, that is not a matter that the periphery acts on the centers through the agency of nerve fibers which reach the periphery, but is some humoral agent which is taken out, which is released from, in this case, from the tumor, which reach the sensory ganglia or sympathetic, particularly sympathetic, through the circulation. So this was the discovery which I described in New York Academy of Science, December 1952. Later on, with Victor Hamburger, we repeated and enlarged on this new discovery. Came out the paper, 1953, Levi-Montalcini, Victor Hamburger, where I repeated what I had already said, but in more extensive way, in the article which I presented as the New York Academy of Science, that gave absolutely evidence that the tumor released by humoral way, not through the nerve fibers, a factor which reach all the cells which are receptive to this factor. All the experiments I mentioned require tremendous amount of time. The chick embryo which had a tumor died in large number. The one which survived were in a difficult condition to study, and many of them also the CAL technique did not work well. So it was taking months to have a result. So I decided that being also very competent in tissue culture, I could study this phenomenon in tissue culture. I say, with the embryo we will take years before we know what is the substance which is released by the tumor. I asked the permission to Victor Hamburger to go and join a friend of mine, Herta Meyer, a German who had been living in Germany during the Nazi period and was working with Giuseppe Levi and myself in Turin. She had established a tissue culture laboratory in the Institute of Biophysics, directed by Professor Carlos Chagas, who became a very close friend of mine. I wrote, with the permission of Victor Hamburger, to Professor Chagas, would he permit us that I go there as a guest of his own laboratory to study in vitro phenomenon which I am not able really to study in detail in the embryo, but I need the tissue culture facility you have and we don't have. To tell the truth, I could have established laboratories there in Washington University because I knew very well about, but I was very curious of Brazil and I wanted to take this occasion to visit South America, so it was an excuse. Anyway, Professor Chagas was delighted at the idea that I joined him, and he did not know exactly what was the reason for. I left St. Louis, I went to visit my family in Turin, and left in October 1952 for Brazil, before the paper 1953 came out, and left and arrived in Brazil, became a guest of Professor Carlos Chagas. And there I made the experiment which became known all over the world, and you have there a photo of the result. I took with me in my pocket 2 mice. At the time people did not look in your pocket as they do now, you know. So I had 2 mice, each one carry a fragment of tumor, mouse sarcoma, 180 or 37 are practically the same tumor, and no one looked what I had in my pocket. Well, when I arrived there, I took away the tumor from the mice and tried to find out if in tissue culture the sensory and sympathetic ganglia adjacent but not close to the tumor which I took from the mouse would grow fibers, and it was a failure and I was in despair because the tumor did not have any effect. So I decided perhaps the tumor must be adjusted to the chick embryo before being studied in tissue culture. I transplanted the tumor from one mouse to a chick embryo, from a chick embryo to another, and then when the tumor grew rather well, I took the tumor which had been cultured, which had been transplanted from the mouse to the chick embryo, and there I get this marvelous effect. That is, the tumor which had been grown for, and still I don't know why, for some time in the chick embryo, produced the effect which is now known as the halo effect, and is described in my book and many articles. That is, sensory and sympathetic ganglia from 8-day chick embryo implanted in tissue culture in the hanging drop technique as we used at the time, adjacent, close to but not adjacent to the fragment of tumor which I had taken from the chick embryo, grew a tremendous number of nerve fibers which became known as the halo effect. That is, in 6 to 10 hours, the sensory and sympathetic cells into the ganglia start to produce a tremendous number of nerve fibers. And I was delighted of this effect because I realized that now I could know about the nature, biochemical nature of this tumor, which I could not possibly find if I continued to work in the chick embryo, you know. So after some period of doubt, and I wrote a long number of letters to Vito, was delighted about this effect, I went back to St. Louis. It was February 1953. Working with Stanley Cohen, we repeated the study which I've done in Rio de Janeiro. He was a young man, 32 years old, as brilliant and modest person, as you can see. And he immediately accepted, when he knew about the result I brought with me from Rio de Janeiro, that it was a fantastic story. So he was very happy to come and accept Victor's invitation to come not as an associate, but just simply as a chemist working with me without a position. And there it happened. Then working together with me and just joining every day the seminar of Arthur Comber, he mentions this effect. He mentions that we did not know if it was a nucleoprotein or what was it. He did not know anything about it, but just we thought a nucleoprotein. And there he received the suggestion from Arthur Comber to add to the medium in tissue culture a snake venom particle which contained the enzyme phosphodiesterase, which is known to degrade the nucleic acid. Therefore, Stanley Cohen accepted the suggestion of Arthur Comber, and he added into the tissue culture a very little amount of snake venom, and just did not tell me anything about it. The following morning, I went there very early in the laboratory, and I could not believe in what I saw. I didn't know what the Stanley Cohen had done. The number of fibers was 1,000 more denser than normal. I said, "What did you do, Stan? Why we have?" Well, he said, "I added a little amount of snake venom, and the phosphodiesterase degraded the nucleic acid. So we know by sure that the effect is due to a protein, and that is not only found in the mouse tumor, but is present 1,000 times more into the snake venom than in the tumor itself." So we knew that the tumor itself produced the growth factor, and that the factor was possibly a protein, not a nucleoprotein, because with phosphodiesterase we had destroyed the nucleic acid. So we knew two things. First, it was not a virus because it was a protein and not a nucleoprotein. Second, it was not a specific only of the mouse tumor, but also was present in very large amount in the snake venom. And that started the 1956 article, which Victor Ember also took place in his, his name is on this article, where we, for the first time, called this particular agent a growth promoting factor. NGF became later on, the P went out, and we thought it was a protein, and Stanley Cohen also identified this protein through the technique, rather root technique at that time, as a 44,000 molecular weight protein. As the first discovery, which I believe was really quite important, I must say, and as a matter of fact, this was the reason for receiving the Nobel Prize, was the first time that it was found a protein molecule, and the protein molecule we know exactly is a source, and also the chemical nature and the gene which produce it, for the first time gave us the idea that the development of the nervous system at that time depend from this particular molecule, which are synthesized in the target cells, in this particular, say, in peripheral tissue. And for a long time, for 30 years, this was the role of this particular molecule, considered to be the first well-identified neurotrophic factor. Others were later on found, and this was the reason they were the precursor of oncogene. For this reason, we received, Stan and myself, the Nobel Prize. Recently, entirely new idea this was never falsified, on the contrary, has been confirmed, this first, but this was the top of an iceberg. The old part of the iceberg was behind the water. What we are finding now is that besides this neurotrophic effect, NGF display other functions which are even perhaps more important than the one we discovered in 1950. That is, this particular protein factor is not only neurotrophin, but is also cytokine. It makes this interaction with between different homeostatic system, endocrine, immune, and nervous system, and perhaps it will be of greater value in all diseases which have to do with deregulation of the system, for instance, autoimmune diseases and inflammation. So without having falsified, on the contrary, confirmed as the first discovery, this was only a part of the function of NGF. Now we see NGF as a neurotrophin, and other neurotrophin have been found similar to, but we also know that this particular agent belongs to this large class of humoral transmitter, which are known as cytokine. It was in 1977, but not even with the 1977 article with Louisa, we realized the importance of the NGF effect on a cell belonging to the immune system, the mast cell. It was only, I must say, 1990 which I realized all this. I mean, little by little, this piece of information did not fit in a scheme, and we just were looking all these without seeing the interaction, which was seen only, I must say, 1990. My 1990 article, for the first time, the article has the title, A New Role for the Neurogrowth Factor. And this was the time, 4 years after the Nobel Prize, where all pieces came together and they realized that the action of NGF is very widespread, much more than we understood before. Before there were pieces of information which we did not really try to put together, not in the article you mentioned, 1968. Before our work, before 1977, the mast cells were considered like a Cinderella into the immune system, coming into action when you have invasion by some organism, and the organism has to undergo hypersensitivity and anaphylaxis. So the mast cell had not been given a very important function in the immune system. The immune system really is the prima donna were lymphocyte B and lymphocyte T. And the mast cells were quite ignored, were found to be active only during hypersensitivity or defense against foreign organisms. When I found out, and this was with Louisa in 1977, that NGF act on mast cells, this was not, had no sense about this, at that time, knowledge of the function of the mast cell. It came about only later, and particularly in the recent years, that the mast cells have a very important function in inflammatory processes. They are very close to the terminal of sensory cells, P cells, sensory cells, nociceptive cells, cells we respond to analgesic stimuli. The mast cell became, for the first time, well, I must say, became if not a prima donna, we realize a function which is not only in hypersensitivity and anaphylaxis, but this cell make a linkage between the nervous system, the immune system, and also endocrine system. They have a new and very important function, and they are in turn very receptive to neurogrowth factor. They synthesize the NGF is an article by Alberto Leon, my name is also on, and the same cells release a large number of explosive cytokines like tumor necrosis factor and lymphokine 1B, and also neurogrowth factor. So the mast cells have very high functional activity in inflammatory processes, and they are under the activity of the immune action. Yeah, you see that a particular immune GIG, AG, and again under the action of NGF. In other words, this cell have a lot of receptors to the immune system and to the nervous system. They are activated not only by these different humoral agents, but they in turn release, synthesize the neurogrowth factor. They release it into the circulation, which has an action in the accumulation local of lymphocyte and other cells, or what you find in inflammatory processes. And this is how we saw right now the mast cell into entirely different role as it was before this discovery. The mast cells are very closely linked to the neurogrowth factor, and in turn also with the immune system and endocrine system. My name is in because we have made contribution, but I wish that it's clear that this particularly the group, Life Group, a research Life Group of Padua, who really worked in great detail these. The article is also with my name because we had a collaboration. But the finding that the mast cell not only have a receptor for the neurogrowth factor, but synthesize the NGF and release it, and this has a very explosive action, cytolesive action, just brought about in the, clearly that these cells have a very, very important multifunctional activity. that are not only there to protect the organism against foreign body or foreign organism, but they release these, and the NGF is a main role in all these. Not only the cell, mast cell, have a receptor track, the high level receptors, the track, but at the same time, they synthesize the nerve growth factor. They release the nerve growth factor together with other cytology like tumor necrosis factor. They reuptake it and have a really very, very central role in all inflammatory process of autoimmune processes or inflammatory process of different nature and neurogenic nature. I mean, the NGF modulates the interaction of the three homeostatic system. But to some extent, it's not only modulating, it brings about all defense mechanisms in activation by alerting the organism that something is going wrong, and therefore the level of NGF increases before the level of the other lymphokine. In this way, we consider not only that NGF is modulating the interaction of the homeostatic endocrine, nervous endocrine and immune, but to some extent there's also an action like, how would you say, a key which opens, which starts the entire phenomenon, you see. I mean, it's the first one to come into action when it is a danger. It's not only that bring about a linkage between all the system, but it's the very first to be increased in amount when something is to alert the organism that something may go wrong. That in 1990, I remember very well, it was during the night it occurred to me, why should be so much NGF into an exocrine gland, which is a mouth salivary gland? Up to that time, nobody had really tried to understand why, because the salivary gland is a typical exocrine gland, and male and female mice have equal need for a gland which has to do with digestive function. Now, at the time, I realized that it was very curious, queer, I must say, that the male mouse should have much more NGF, 10 times more NGF in his own mouth salivary gland than the female, because both have the same need for digestive function. Then it occurred to me also that mice in the wild live in groups together and are very aggressive against each other. So I thought that perhaps this large amount of NGF has some different functions from the digestive function. And perhaps this was the reason why male and not female has so much more. Male are more aggressive than female. And immediately I made the experiment, which proved the reality of this, to put male mice, adult male mice together in isolation. This was it was a kind of treatment which had been discovered, or at least realized by Valzelli, a very good, now he passed away later on, young man, that he decided to study aggression in mice by isolating from each other, then putting them together in a cage. When they have been isolated for 2, 3, 6 weeks and are put together, are very, very aggressive. Now, during aggression, I decided to find out what happened to NGF. We studied the NGF into the circulation, not close to the salivary gland, in order not to contaminate, and found it was loaded with NGF. This occurred to me in this particular time, that this was the reason why the mouse salivary gland, male mouse, has so much NGF. It is nothing to do with digestion, but with a defense mechanism, particularly male against male. You understand, sir? This to me was a very important discovery, and it was later on proved also in men. As you remember, it was Louisa work in this laboratory 3 years ago, that the young people, soldiers, which are due for parachute experiments the following day, in the day before, the more NGF into the blood normally is sub-threshold amount. But when a young man, 20 years old, is being told that the following day will be for the first time parachuted, the NGF increase in his blood due to anxiety. In the following day, when the same young man, or many of them, there are 50 or 100 were studied, reach the floor, the parachute stop, they took the blood, Louisa and other people on my group, and found it was loaded even more than the day before with NGF. So the aggression which we have discovered and had the idea of this experiment into the male mice has equal kind of result in a human being parachuted. Well, to some extent, it shows that emotion, any kind of emotion, not only aggression itself, are very important for the release in circulation of NGF as a defensive mechanism, the subordinate mouse, or the anxiety of the young man who the following day will be parachuted, is a way to activate the NGF into the circulation, which in turn bring about activation of lymphokine, which are defensive. You know, I don't know if I'm clear enough, but anxiety is an additional phenomenon or additional state, event, which increases the amount of NGF. Is not only the aggression itself, which is just the way how a mouse attacks another mouse or a human being, but it is the state of anxiety which shows that NGF works not only, as we know, about differentiation of different systems, but also on the common in the behavioral of the person. When the person is very anxious, and this is also true in diseases, this increases the amount of NGF, and in turn the NGF bring about activation of the lymphokine. I mentioned that when a young soldier for the first time is called to parachute, and as first anxiety and then the activity, the traumatic action of the parachute, the NGF growth the level of NGF increase into the blood. But this precedes the increasing level of lymphokine, which also act, which are activated. So this gives the idea, and we are in favor of an alert phenomenon. The fact that NGF increase before any increase of other lymphokine, which are also in the circulation, show that the action of NGF is preceding a sort of alert when it is a danger, when it is anxiety or other things. First is the synthesis of NGF, then this in turn brings about the increase of lymphokine. And this came particularly after the parachute experiment. If I have to say, the people who work with me or not with me, but made major contribution in the study of the nerve growth factor and other growth factors, certainly the first to be mentioned is Stanley Cohen, who, working with me, discovered the nature of the nerve growth factor, and immediately after this discovery, discovered the epidermal growth factor, and for this reason he received the award. It was given to me for the discovery of the nerve growth factor. To him we shared, of course, it for the discovery of the epidermal growth factor. But other people also made outstanding contributions, and I wish to remember that already, I believe in '76, Lloyd Green, a young man at that time, and still young today, I believe, not so young as he was at that time, made a very, very important new modern discovery, open a new development to the nerve growth factor by finding out that cells, malignant cells, originating from chromaffin cells, so-called pheochromocytoma malignant cells, respond in vitro to the nerve growth factor by changing from a type of endocrine cell to a nerve cell. This model has been used ever since its discovery up to today up to the present day to find out the mechanism of action of nerve growth factor at the cellular and molecular level. Following this, and still today we use this system discovered by Lloyd Green, other people made outstanding contribution. The one which two particularly worked together and did a large amount of work is Hans Thönen from Switzerland, who did really very, very important work on the study, some paper with us and some by himself alone, how not only the nerve growth factor act, but other brain-derived growth factor act. And so Stanley Cohen made, Hans Thönen made a large study, and very important, still today continuing, on other growth factor, neurotrophic factor, ciliary factor and others. Finally, a person, and particularly fond and admirer of, is Erich Schutter. Erich Schutter entered in this field in the early 60s. He came from England and went to Stanford, where he is still now, and his work is magnificent from the point of view being a chemist, knowledge of the way how the NGF works, and particularly through the discovery of the way it works on a receptor. He was the one who discovered the two receptors, and with other people more recent, Chow and Kaplan and others, made very, very important study and really opened new way by finding out how this low and high activity receptor P375 and TREC work in the bringing about the effect of the NGF inside the cells. Well, these are the one which I want to mention, but naturally I have to mention the people working with me, particularly Pietro Calzano and Luigi Aloe, a very close friend of mine, who in these last 30 years have been working with me all the time, and they did a lot of work. Then I mentioned the Life Research Group of Padua. I am very close to them, and we have been working particularly on the immune system and particularly on the action on mast cells. My first mentor was Professor Giuseppe Levi, a man of great stature as a scientist, as a human being. Three of us at the Nobel Prize, we were schoolmates, Luria, Salvador Luria, Renato Dulbecco and myself. We were in the same class, and we all were admirers of Giuseppe Levi, even if no one of us followed the real path. He was an histologist, and he did not make a very important contribution scientific, but it was the personality of Levi which impressed all of us. Also was a pioneer of tissue culture in Italy, very much more in cultural man and than all his colleagues. And so I have a tremendous recognition, gratitude for him, for what he gave to me, for the way he acted. He was a very, very violent anti-fascist. He went to jail, and he was very, very courageous human being and ethically of high level. The second person, my mentor, Victor Hamburger, to some extent, as a personality, is the opposite of Giuseppe Levi. Giuseppe Levi was temperamental, violent, and difficult. Victor Hamburger, a very gentle person, highly cultural person, and very pleasant to work with him. We had an excellent relationship, and I have a very lot of gratitude for having brought me to Washington University for the friendship which was established. As a matter of fact, I dedicated to him the book which will appear in February in the Singapore, which is the 20th century Nobel Prize winner. Each one of us wrote a volume. My volume is the saga on nerve growth factor I dedicated to Victor Hamburger. These are the two mentors who had a great influence on my life from the point of friendship and different way of approaching life. The Vatican Academy, I am really proud of being the first woman nominated by Paul VI in 1974 as a member of the Academy, has done excellent work on different scientific problems and projects, always was very open-minded and never just related to religious feeling, because many are Jewish members of the Academy. But besides the religion, I mean, it's a very open way to look to scientific problem as related or not related to religion. And as I say, I'm very proud, and I consider the work, particularly this year also, was excellent work, which has been done in different fields of physics particularly. So the contribution of the Vatican Academy is very, very important to science and to related of other problems, social problems, religious problems, and scientific. The religious feeling is something which is inside of Homo sapiens, the need to believe or not to believe. I am a highly ethical person, I believe. I give very importance to the way I will live. However, I am considered agnostic. I do not really belong to any particular group, but I have a high respect for all of them, Catholic, Jewish, or different kind. I believe that in the Vatican is very much respected, this attitude. I made clear when I was nominated on my positions, they say, we don't care about it. We care about you as a scientist and a very highly moral person. And I very much liked and enjoyed this way of looking to me. As a child, I was very close to my mother, still up to the day she unfortunately died. Father was a very powerful personality, and I had some problem with him just because I cannot really accept his very powerful personality. After he died, I realized how much I owe to him. He died quite young. My three brothers and sisters are Gino, who was a very famous professor of architecture and science and artist. He died also young of a heart attack in '72. We were very, very close, and I'm very close to his children. The other is Anna, or Nina as we call, the elder sister, still alive, is 92, very lucid in mind, but she has a lot of serious problem. And we speak to each other every day over the phone, or I go to Turin to visit her. My twin sister, of course, is very, very close to me, is Paola. She is a very great artist. Recently she has not done anything. What you see here is her work. She has never been acknowledged as she should have been, because very modest and very indifferent to recognition, too much so, because she is really, she has been a very, very great artist. She was a student of Casorati, one of the most famous Italian artists in the 30s and the 40s. And we are very close, we live together. And this is the reason why I consider myself more as an artist than a scientist. I'm not, well, I know and I have no problem with mathematics, but I'm far away from being in this kind of a very rigorous analytic approach to reality. I'm much more intuition mind, that is the right brain, and holistic way of looking things. And I believe that my discovery in my way just owe to this particular, if you can say, gift which I have, to be very intuitive, to have a very intuitive and artistical mind. All people are not generally considered just out of connection with the world, but this isn't true. It is true that we lost a lot of nerve cells every day, practically 100,000, but the capacity to have this cell working, even in old age, has been proved by people like Russell, like Ross Blotter, the Pugwash founder, and the atomic physics who worked on atomic bomb, by great artists like Michelangelo and Picasso, who were over 90. Russell was 98 when he died. Ross Blotter is 1 year older than I am and very active. So I do believe in order to be creative in old age, at first you have never to consider yourself but just problem, and never be interested in what you are yourself. I'm now 87, you know it, but I never think of it, and I don't care when I will die, if tomorrow, or if I will have more weeks to go or months, I don't care. I do believe that by having the brain working very much, and I have some theory which I will write down, I cannot tell you because it's too long to speak about this, that you can improve your creativity by just working very hard and never thinking of yourself, and also, I must say, to have some particular capacity of having your brain working. But this I will write in a book which I'm in the process of bringing to completion. And the title I mentioned to you in English will be A Winning Card in a Tether at the Sleeve, which comes from beautiful poem by Yeats, Sailing to Byzantium.